Okay, so good afternoon. It's my pleasure today to introduce Dr. Ian Bruce. I realize I've known him since 2022 because that was the year Simon Haken in electrical engineering recruited him to come and join him in his work on hearing aids and how the auditory system works. Ian did his bachelor's at the University of Melbourne, Australia, in electrical engineering. Took a year in Vienna at the University of Technology, and then he did a postdoc at Johns Hopkins University. So he's a good pedigree. He's had many honors, but including his honors are a fellow of the Acoustical Society of America. And Ian, I think he's one of those unusual engineers that he really understands psychology, experimental design. He really knows a lot of neuroscience, and of course he's a very, very competent computational engineer. So his research interests really are all related to understanding and diagnosing and treating hearing disorders. So he's worked on a bunch of areas within that. Of course, at the Live Lab, we're really happy that he's done over quite a few, I don't know how many years now, at least 10 years, work on hearing aid research with the Hamilton Philharmonic and in the Live Lab. He's also an associate member of our department. Very valued associate member of our department. I couldn't think of anything bad to say about him other than he's a really nice guy. So if you don't know him, get to know him. He's always willing to talk to people, and you'll learn a lot when you talk to him. So, Ian.
Great. Thank you, Laurel, for the kind introduction. And I must admit, an attraction to coming McMaster was not just a great engineering faculty and colleagues there, but Laurel Trainer, the work that she was doing at the time, and since some that I've been able to collaborate with her. Sue Becker, who gave the talk last week, I had several collaborations with her, and then a lot with the late Professor Larry Roberts when he was doing tinnitus research here. And then as an associate member, I've been on quite a few supervisory committees for other people doing auditory or related research in PNB. So I've really enjoyed my connection with PNB, and it's a real honor to be able to give the colloquium today. So as Laura said, my research is fairly diverse, from spanning computational and experimental approaches, and some very applied hearing aid and cochlear implant research. A lot of what I've done since my PhD has been computational modeling to try to create a link between what we know about physiology, auditory physiology, and auditory perception. And we use that as a tool in a lot of this applied work that we're doing. But it also provides a lot of insight, I think, into how auditory perception works. So this is a bit of an overview talk, talking about some older work, some development of the models that we have in my lab, some applications that we have in my lab, and then some applications that some other groups have been doing for this modeling work. So to answer this question of what the ear tells the brain and why, I'm going to start out a bit with the challenge of auditory perception. So if you think about that in a listening task, we have auditory perception in our daily life. We're getting sound pressure waves that are arriving at each eardrum, and somehow the ear and brain have to make sense of that auditory scene. And the renowned auditory psychologist Al Bregman from McGill University, in his textbook on auditory scene analysis, he likened this to somebody trying to understand what was happening out in a bay with different types of boats that might be out there, people swimming, different animal life that's in the bay, trying to understand what's going on there, just based on the patterns of vibration that you could get if you were to put a couple of channels into the shore and put a handkerchief over each of those channels and just watch the handkerchiefs go up and down with the wave vibrations. That this is an incredible task to be able to take these one-dimensional time pressure waveforms and somehow analyze what's happening out there in the auditory world. So the first answer to what the ear tells the brain is that it's not trying to faithfully represent those pressure waveforms over time up to our limit of hearing, say in humans 20 kilohertz. If you're in the bat lab, up to more like 80, 90 kilohertz. That's not really feasible to do physiologically. And so what happens is that there is a time frequency analysis that is happening in the cochlea that is helping the brain out in not having to deal with these very high frequency pressure waveforms that are arriving at the eardrum. So all the way back in the mid-1800s, Helmholtz, based on some observations from anatomists at the time, thought that the basal membrane in the cochlea could be performing time frequency analysis via tuned resonances along its length. So anatomists had taken sections out of the cochlea in the ear and observed as you go from the base up to the apex that even though the snail shell-like structure of the cochlea is getting narrower, the basilar membrane is actually quite narrow and stiff at the base and becoming broader and less stiff up at the apex, and had the idea that these could be tuned resonances then that when a wave comes in through the stapes from the middle ear, it would set up vibrations, and the mechanical tuning of the basilar membrane could then do a time frequency analysis, and that then, the output of that could then be sent off to the brain. So it's not having to try to represent the entire pressure waveform. Now, an issue with time frequency analysis is that there's always a time frequency trade-off. So people here may have some familiarity with the Fourier transform as one of the most widely used forms of frequency analysis. If we want to do this over time, then this is referred to as the short-term Fourier transform, STFT. And there's an inherent trade-off that if you want to have small time windows over which you're analyzing the spectrum of a signal, such as a sound, then the frequency resolution is not very good. We have broad frequency representation. If instead we want to have a good frequency representation, instead we need to have long time windows. So you can't have both. There's a time frequency trade-off. So if we were to look, first of all, at the pressure waveform of a speech signal, in this case shown up here, and to do the short-term Fourier transform and now plot the magnitude of that as a function of frequency and time, if you have the short time bins, you can see that the frequency information gets smeared out over frequency, but we've got very good time representation. Say here, these transient parts of speech are well represented. Or we can have a poor time resolution, but a very good frequency resolution. So down here in this narrowband spectrogram, we call it, because it's got narrow frequency representation. Now we can see the harmonics nicely represented for the vowels here. But these transients that are occurring in the consonant parts of speech here are smeared out in time. So this is what we get if we're using the short-term Fourier transform that has this linear frequency scale where we have equally spaced center frequencies for our frequency analysis, and the bandwidth is constant along the length. But if you're designing a filter bank, you don't have to have this linear frequency scale. You could have a nonlinear frequency scale, one, for example, where the center frequencies of the filters are now octaves of each filter. So we're going from our base frequency here to 2 times, 4 times, 8 times. And in this case, the bandwidth of the filters are also getting broader as we go. So we're covering the full spectrum. So now if we think of it in terms of the spectrogram, frequency and time, we've got good frequency resolution, poor time resolution for low frequencies, and then as we go up in frequency, we're getting better time resolution, but at the cost of frequency resolution. And if you think about what I just showed in the spectrograms for speech, this might work fairly well if the vowels are mainly these long, low frequency utterances that have interesting harmonic information. We want good spectral resolution. They're fairly long, so we don't need such good temporal resolution. But for the transient parts of speech, maybe we want better time resolution, and we're willing to give up frequency resolution. So what frequency scale does the cochlea have? So it took almost 100 years before someone was able to take a cochlea, in this case a human cadaveric cochlea, and do vibration measures to start work out what the actual mechanical tuning was. And if the measurements were done at different places along the length of the basilar membrane, here fairly equally spaced, so if you look at the numbers, they're 3 to 4 millimeters between each of these measurements, you can see that the relative tuning that we get at each place on the basilar membrane as a function of frequency looks fairly constant, equally spaced, fairly constant bandwidth, until we look down at the frequency scale here and we see that this is actually a logarithmic frequency scale, not a linear frequency scale. So it turns out that, at least from these cadaveric recordings, it looks like that the frequency scale along the cochlea is closer to logarithmic than linear. So we have a better frequency resolution at low frequencies, and it gets poorer as you go to higher frequencies. Now this is in cadaveric cochlea. It's possible that things change post-mortem, and we'll see in a bit that they actually do. There are psychophysical methods people have used to try to estimate this place to frequency mapping in the cochlea, and I've shown several of these curves here in the colored line. So this is the normalized scales or normalized length along the cochlea versus frequency now on a linear scale. The black curve is showing a logarithmic scale, and these different psychophysical estimates you can see are close to log, but they're slightly more linear than a log scale. And there's newer estimates from basilar membrane recordings that suggest that these are pretty good estimates. There's some species differences, but if we're thinking about mammals in general, this is sort of a fairly good frequency scale that we might have. And just to note here, I'll talk about sort of the usefulness of this type of frequency scale versus alternatives such as a linear frequency scale. This Mel frequency scale is one that's used a lot in applications like automatic speech recognition, so using machine learning to do tasks like humans. That's been found in that case that using a cochlea-like frequency tuning seems to work better than using alternative, and particularly this Mel scale is quite widely used. And so that brings up a question of is this somehow optimal some way? But first of all, want to talk a little bit, I've been talking about spacing of a frequency along the cochlea. So one issue in these estimates is that the cochlea is really continuous. we have discrete inner hair cells that are taking the vibrations of the basal membrane and turning that into excitation of auditory nerve fibers. So it is discretized at some stage, but the basal membrane is continuous, and so we're just sampling it at discrete places. And so what matters a little more is not just those center frequencies, but also how sharp the tuning is as we go along the length. And the way that engineers quantify this sharpness of tuning is through a quality measure, so it's called Q, and it's the filter center frequency divided by its bandwidth. So if we have an example filter here, we need to define the bandwidth somewhere. So one common one that you'll see in some upcoming slides is to say, what's the bandwidth 10 decibels down from the peak gain of that filter? We'll define that as the bandwidth, and we'll find the center frequency of the filter, divide the center frequency by that bandwidth, and if we've done it at a particular level, say at 10 dB below, then we'll call that the Q10 value for that filter. So if we go back to these example filter banks that I showed, so our linear frequency scale that we have for the Fourier transform, that ends up being a constant bandwidth filter bank. So we have equally spaced frequencies, and the bandwidth is constant as we go along. And this nonlinear frequency scale here, we saw that the center frequencies go up by octaves, so doublings, but the bandwidth does as well. And so when we divide the center frequency by the bandwidth, the Q value stays constant now at different center frequencies. So these are kind of two different possibilities, potential extremes that we could have in terms of the cochlear filtering scales. So it looks like that it's not quite logarithmic, that it's somewhere in between constant bandwidth and constant Q. And the question is, why is that? Is there something optimal about having filtering that's between constant bandwidth and constant Q? So Mike Lewicki, researcher who solved for a bank of 128 filters that optimize coding efficiency. So he took digital filters and had an algorithm that optimized how information was encoded through those filters that effectively is minimizing redundancy statistically in the outputs of each of those filters. And then he used databases of different types of natural sounds to come up with different sets of optimal filters. So the main results of his paper are shown here in this figure, and these are plots then of the frequency versus time representations. So he's done it here as ellipses rather than breaking it up into rectangles like in the previous plots, but it's the same idea. And the question is, well, for what type of sounds do you get these different shapes of the filters? So it turns out that animal vocalizations, was actually mammalian vocalizations, so these are mainly vowel-like utterances that have a strong harmonic structure, that it's optimal to have these very long, sort of poor time resolution, but good frequency resolution for the filtering of the animal vocalizations. If instead you train this algorithm on environmental sounds, you get some very long and narrow filters for low frequencies. That makes sense. There are some environmental sounds that are kind of resonances, where you get things that are happening at low frequencies and going on for a long time. But a lot more environmental sounds are very transient, and so up at the high frequencies, you get more of these broad frequency representations, short temporal representation. And then if you train this algorithm on speech, you get something that's in between. So you get more mid-frequencies that have have long temporal resolution, but good frequency resolution, and then you have to go to the higher frequencies before you start getting more of these ones that have poor frequency resolution but better time resolution. So there's some diversity, but that's the general trend. It's a little hard to sort of work out how strong those trends are from these plots. So what he then did was quantify it in terms of the bandwidth and then these Q10 values as a function of center frequency. So you can see you get nice separation for the three different types of sounds. So the environmental sounds, you get a bandwidth that's increasing with center frequency. Speech is less of a growth in bandwidth. And then the animal vocalizations have constant bandwidth. So for animal vocalizations, the short-term Fourier transform would be optimal. If we then think in terms of the Q10, so now a higher value means a sharper filter, we can see that the environmental sounds now have the lowest Q10 values. It's not quite constant Q, you're getting a slight increase in the Q10 value with center frequency, but not a very strong growth. If you go to speech, you have a sharper growth, and when you go to the animal vocalizations, you get the steepest growth. And plotted here with this, it might be a little tricky to see, but this solid and dashed line here, these are estimates of the Q10 versus center frequency from mammalian cochlea. Now, there's some differences across different species of mammals. There's evidence that human Q10 versus center frequency might be a little higher, so a little sharper than smaller mammals, but the slope is very similar across mammals. Now, it might seem from an evolutionary point of view, why would a cat's or dog's ears cochlear tuning be matched to speech? Lewicki then said, well, what if I were to train these networks on a mix of animal vocalizations and environmental sounds, which is what animals might really be experiencing. And he found that if you had a 2-to-1 mix of environmental sounds and vocalizations, and you train the filters on that, you end up with something that's very like speech. So he argues from an evolutionary point of view, being able to well represent speech sounds and environmental sounds is important, and then human speech has developed to optimally make use of the tuning that the mammalian cochlea has. So up until now, I've been talking about these filters and the scale, so the frequency spacing and bandwidth of these filters. Importantly, after the work of von Beckersay, further measurements that were done in live cochlea showed that the tuning is actually nonlinear. So if you do a measurement at one place in the cochlea and look at how the vibration growth, how the vibration grows as a function of the sound pressure level of the acoustic waveform, we can see that the growth can be nonlinear, so compressive in this case. And if we do plot it instead as the effective gain, so the amount of displacement per Pascal for the sound wave that we're putting on as a function of frequency now with each curve corresponding to a different sound pressure level, we can see that at low sound pressure levels, there's this very sharp frequency tuning and high gain, and then as the sound pressure level increases, that the gain gets turned down, and the dashed line is showing the post-mortem tuning. So there's some kind of active mechanism in the cochlea that's amplifying quiet sounds and making the basilar membrane tuning sharper. We now know that's due to the outer hair cells in the organ of Corti within the cochlea. So this gives rise to this kind of compressive behavior for frequencies that are near this best frequency or characteristic frequency for that point on the basilar membrane. There's also suppression effects that can happen if we've got more than one sound frequency at a time. So this is showing what happens if you record from an auditory nerve fiber and measure the threshold of it responding to sound as a function of frequency. This open circles are indicating here for this particular auditory nerve fiber the threshold of sounds of different frequency that are required to excite them. So this is the excitatory frequency threshold tuning curve here. But if you then play a sound that's at this called a probe tone here that's inside the excitatory tuning curve, so that'll make the auditory nerve fiber respond more than it will spontaneously, and then you introduce a second tone of a different frequency and amplitude, you can find these regions of suppression where if you add a second tone in, that'll actually suppress or reduce the auditory nerve firing. So there's some type of suppression or contrast enhancement that's occurring through this outer hair cell control of the basilar membrane vibrations. So overall, these nonlinearities, we now know that part of what they're doing is helping with the very large dynamic range of hearing. If we listen over a range of about 120 decibels from the quieter sound, we can hear up to the louder sound that's going to start causing us pain and hearing loss. That's a huge range. This compression helps our ear and brain deal with that somewhat. I'll show some evidence that it also helps stabilize the representation of speech at conversational levels, and it provides some contrast enhancement. Again, I'll show some direct evidence of that in predictions. Another nonlinearity that's important for perception is that different auditory nerve fibers, if we look at fibers that are innervating different parts along the cochlea, so they have different characteristic frequencies, at any one point, different auditory nerve fibers could have different thresholds at which they start responding to sound. And you can see this region down here, the absolute threshold, that's saying if a sound is quieter than that, then no nerve fibers are going to respond, the sound is going to be inaudible, and then as you increase the sound level, you're going to recruit more and more auditory nerve fibers, so the sound's going to become more and more audible, easier to hear as the sound pressure level increases. So this thresholding behavior for quiet sounds has a direct perceptual effect that will come into play in some of the results that I'm going to show. And then the next bit of important information about how the cochlea, the hair cells, and the auditory nerve fibers work is that auditory nerve fibers are able to synchronize to low frequencies, so below a few kilohertz. So if we have a sound that's, say, at 1.1 kilohertz, and we do a recording from auditory nerve fibers and count the numbers of spikes that are occurring as a function of the phase of that sinusoid, we can see that spikes tend to preferentially occur at a certain phase. We can't have negative spikes, so for the negative phase of the stimulus, we're just getting suppression of spiking. And you can see that we have a fairly stable representation as we're going up in sound pressure level here, that this phase locking or synchronous representation of information is fairly good over a range of sound levels. Importantly, it's been shown here from the seminal work of Don Johnson. So I mentioned this strong synchrony. So if we quantify how strong this synchrony is, here is a synchronization index. We only get strong synchrony at high frequencies, and at about 1 kilohertz, it starts falling off, and we don't get good synchrony information above a few kilohertz. So that's going to play into some of the perceptual results that I'm going to show. So it's one thing to look at this different physiological data from all these different things and then to say, well, how's that going to relate to perception? And psychophysicists, people studying speech perception had to do that for a long time. But then computational methods and computational resources became available that we could start to formalize our understanding. So in the series of models that I've been working on, the first version was from Laurel Carney's lab, first published back in '93, where she had a section that was modeling the basilar membrane filtering, the inner hair cell transduction, hair cell adaptation, and discharge generation in a single auditory nerve fiber. And then by changing the center frequency of this, we could model a whole population of auditory nerve fibers. This was one of the first attempts to try to introduce the cochlear nonlinearity. She used this feedback mechanism from the basilar membrane filter to introduce the compression behavior, but that couldn't then explain that off-frequency suppression behavior. So this is when I started working with her. I was doing my postdoc in Baltimore, and we met at a conference and started working together, and we changed it so that now this outer hair cell control path was a feed-forward mechanism to the signal path filtering, so the basilar membrane vibration that's driving the inner hair cells, but as feedback onto itself. And we found this alternative structure. We could explain both the on-frequency compression and the off-frequency suppression behavior by having a wider filter for this control path for the nonlinearities. I subsequently, with my postdoctoral supervisors, Murray Sachs and Eric Young, did some further development. We were interested in explaining speech perception and effects of hearing loss on speech perception. We found for broadband stimuli like speech, we needed to include the effects of middle ear filtering, which is kind of a broad bandpass filtering, and did some changes to the control path to minimize some distortions in it, and then added some factors where we could introduce inner hair cell and or outer hair cell dysfunction to induce hearing loss, and then use it to study the effects of hearing loss and hearing aid amplification schemes. In parallel, Laurel Carney's lab was developing some improvements to the main model of the basilar membrane filtering here, and then back in 2006, I had a PhD student, Mohammed Salani, who incorporated Laurel Carney's lab's improved model here, and also we found we had to add an extra filter path up here to explain what was happening for very high sound pressure levels in the auditory nerve data. And then over the years, we've done some further improvements to the synapse model and spike generation parts of the model that I won't go into here. They're not crucial for the results that I'm going to show. So there's lots of applications of this model. So we have used it for evaluating and designing hearing aid amplification schemes, predicting central effects of hearing loss, and designing tinnitus treatment. So again, a lot of these done with colleagues here in PNB. Also used it to do some prediction of music perception, speech intelligibility and quality, and in some other labs, I'll mention the data, predicting performance on psychophysical tasks. So I don't have time to go through everything, so I'm going to give one example from each of these final three applications here. So the first is one they actually did with Laurel just over 10 years ago. It was explaining the high voice superiority effect in polyphonic music. So why is it that the higher voiced instruments tend to get the melody, the sopranos tend to sing the melody in a choral piece? So why is that we like to have the higher voiced instrument or voice carry that melody? Why is that pitch more salient? So Laurel with Celine Marie had some EEG mismatch negativity data where they were playing synthetic piano tunes simultaneously and in an oddball paradigm having a deviant where the higher tone could go up or down or the lower tone could go up or down. And they found, looking at these difference waveforms, that whenever there were two tones being played, if there was a change in the high tone, it gave a bigger mismatch negativity than changing the low tone. And so Lowell came to me with an interest of could the model explain how this worked. At the time, Gavin Biddleman, who'd been doing some modeling of music perception with the auditory model in his PhD at Purdue University. He was doing a postdoc at the Rotman Institute, so we banded together and worked on using the model to predict this data. So one of the metrics that we were using to have a neural saliency measure was making use of this spike timing representation of low frequency information. So what we were doing was pulling interspike interval histograms, which is one way of representing the timing information in the auditory nerve. So pulling that across a population of neurons, and then applying this harmonic sieve. It's basically looking for are there intervals that correspond to a particular pitch period, or its integer multiples that might represent that particular pitch. And then if you change the pitch of that harmonic sieve, you can see how much of the synchrony information in the neural response is representing tones of different frequencies. And you can see in this example here, a two-tone case, the upper tone salience is predicted to be substantially more than the lower tone salience. And in general, this is the case. So this is showing in red upper tone neural salience, blue lower tone salience, where we're starting out with the two at the same frequency and then changing the frequency of the upper tone. So we can see as we have a upper tone frequency that moves away from the lower tone, we get this much higher salience here, and the ratio of the two is represented on the right. So that's true in the lower musical register. As you go to higher and higher musical register, that difference tends to go away, and that's understandable given that, again, that the temporal representation has a cutoff of a few kilohertz, so this is not too surprising. He showed, Gavin Biddleman ran some simulations with an example here just looking at the soprano and bass parts in Bach polyphonic vocal piece here, Bach chorale, and showed, again, for this real listening case here, that the upper tone salience predicted by the model is normally higher than the lower tone salience. Now, a question might be, what is it about the cochlear processing that is causing this upper tone to be more salient? And if you recall, we said the nonlinear frequency scale, the frequency resolution in terms of the bandwidth is actually better at low frequencies than high frequencies. So that was one of the kind of questions that Lowell came to us with, saying it's kind of backwards. We would expect just from the frequency tuning that the lower frequency would be represented. This neural pitch salience model from the synchrony information seemed to show that no, it's actually the upper tone that's more salient. So we also ran some simulations looking at the place rate representation, so looking at the spike rate along the length of the cochlea. And so what I'm plotting here, blue in this upper panel is the spectrum of the low frequency tone in the mismatch negativity paradigm. This middle panel is the higher pitch tone here, and the green curves are the neural spike count we get out of the model as a function of the center frequency or characteristic frequency of the auditory nerve models. And we can see in isolation we get this nice representation of the lower harmonics of the low pitch sound. We get a nice spatial representation of the harmonics of the higher pitch sound. When we put them together, because of the way that this compression and suppression effects work, and a little bit because of middle ear filtering, attenuating the response to the low frequency component, fundamental frequency. If you look at the green curve here for what you get when they're combined, the shape here actually looks closer to the high tone sound than it does to the low tone. Basically, these harmonics here in the high-pitched sound are suppressing the neural response to the nearby harmonics from the low-frequency sound. So kind of the wider spacing that you get from the higher voiced sound here is helping with this suppression effect. And we did a control case of getting rid of the middle ear filtering and turning off the cochlear nonlinearities. And if we do that, you can see some of the contrast enhancement goes away in this middle case. I'll go back to with cochlear compression and suppression and without. So you can see without this cochlear nonlinearities, you don't get as much contrast enhancement. And you'll also see, if I flick back to with compression and suppression and without, you can see that without it, the neural response to the harmonics in the low frequency sound go up again. So they're more equally represented. So in this case, the cochlear nonlinearities are actually helping the higher pitch sound be better represented than the lower pitch sound when presented together. Another application that we've been looking at for many years in my lab, I'm going to show some results that are hot off the press from my PhD student, Yu-Ji Lee, who is here. So there are effects of sound intelligibility as a function of sound level, and you've experienced this yourself, that people sitting in the back, if I wasn't amplified, you'd have difficulty hearing me. You'd say, oh, a lot of it's inaudible, it's too quiet. But you've also probably experienced, if you've been at a party where there's maybe more than 15, 20 people and you're in different groups and maybe there's some background music playing, that one group will be a little rambunctious and talk loudly and you won't be able to hear the conversation in your group, so you start talking a little bit louder and you can hear each other then and you're hearing it, but then the other groups have to start talking louder and just escalates and escalates. And does talking louder ever help? Does it really work? No, you end up telling people turn the music down. If you're brash, you might tell the other group to not talk so loud. Often people end up leaving and going somewhere quiet outside of the main area of the party. So that's what happens in a noisy situation with lots of conversations. It actually happens even with just one talker. So this is some interesting historical data looking at the perception of nonsense syllables, so percent correct as a function of sound level here. They've got a slightly weird sound level here. 0 on this scale corresponds to conversational speech level, so about 65 dB SPL. So you can see this audibility effect as you increase the sound pressure level, the percent correct goes up and then it plateaus, but then you also get this rollover effect where intelligibility starts going down again if you go above conversational speech level. Now what's being plotted here is if the speech is low pass filtered with different cutoff frequencies, the lower the cutoff frequency here, the more you're getting rid of the mid and high frequency information, the intelligibility gets worse. Similarly, on the right here, if you high pass filter the speech and you move the lower cutoff frequency higher and higher, you're losing more and more low frequency information. Now intelligibility gets worse, but you still get this rollover effect where if you go above conversational speech level, that it gets actually harder to understand the speech even in quiet. So, as mentioned, this is data back from the 1920s. This is interesting data. The reason for doing these different cutoff frequencies, this was data collected at Bell Labs developing the North American landline system of the last century. And based on this data, they decided to filter between 300 hertz and 3 kilohertz, that would give reasonable intelligibility. And they worked out as long as the sound pressure levels close to normal conversational level, people do well, but it wouldn't help to amplify it more. In fact, it could make perception worse, and it also might be uncomfortable to listen to speech that was amplified too much. So how do physiological models go at explaining this data? So I'm showing, first of all, one metric that's the hearing aid speech perception index developed by Cates and Earhart, where they have, I won't go through all the details here, but this is a much simplified version of the auditory periphery model that Jim Cates came up with to try to make it run faster. Our very detailed model's relatively slow. He wanted this to be much faster, so he greatly simplified it. And he has a somewhat complex way of predicting intelligibility, but it's basically looking at the modulations in the neural response, how well they're representing the modulations, temporal modulations in the acoustic information in the speech. And these are some results just coming in from Yuji's work here, so I'll just put the perceptual results up here as a reminder. So shown on the left here are predictions for the different low-pass filters and for different high-pass filter cutoffs, and you can see that this kind of general rollover effect occurs, but the curves are pretty funky, and the effect of the cutoff frequency doesn't really match very well with the data. So we were interested, does our more physiological detailed model do a better job? And so we're taking our model doing the same thing. The first metric we're looking at is called the neurogram similarity measure. So it's looking at the response that we get from clean speech at conversational speech level, and then speech that's been altered in some way. In this case, just the presentation level has been changed relative to conversational speech level. And we're comparing the neural output that we get from the model for those two cases. This metric compares the mean discharge rate or spike count that we're getting in little 3x3 pixel windows, so frequency and time windows, and then averaging. And then it's got this second term that's looking at the correlation in the spike counts between the two neurograms in these little 3x3 windows, and then it averages it over the whole neurogram. So if the NSIM is 0, it says it'll be completely unintelligible. These two neurograms are too different from each other. If it's equal to 1, it's perfectly intelligible. So what do the NSIM predictions look like? At the moment, I'm just doing a linear mapping from the NSIM value to the predictions. And you can see that the curves now are a bit more orderly looking, with level, and the effects of cutoff frequency are a bit more sort of graded and consistent, but this kind of growth of audibility and rollover effects seem to be a bit strong. So it looks like this metric, if I flick back, this term that depends on the overall spike counts of the two neurograms might be a bit strong. So there's an alternative metric that's a bit closer to what the Haspe metric's doing, that's looking at the modulations in the neural representation, this time inspired by models of cortical modulation filtering. So similarly, we're looking at, this case, it's called a template response to the reference, and a noisy neural response to the test signal here. And again, we can mathematically work out a relationship between the two, 0 if it's completely unintelligible, and 1 if it's perfectly intelligible. Again, at this stage, we've just done a linear mapping, and the simulations with all the different cutoff frequencies are still running. We didn't have ready, but we did plot the STMI predictions for the wideband case, so with no filtering, and for one low-pass filtering case and one high-pass filtering case. And here you can see this predictor is better able to predict the stability of the intelligibility over a wider range of sound pressure levels. So it looks like this STMI metric, along with the stability that the cochlear compression and suppression give, might better explain why we have a fairly good intelligibility over a range of sound pressure levels around conversational speech levels, and yet still predict this rollover effect and the intelligibility effect. And I'll just finish up briefly, realize I want to leave some time for questions here. An interesting application of this work from Josh McDermott's lab at MIT, headed up by his former PhD student Mark Sadler, looked at training deep neural networks receiving inputs from a cochlear model on psychophysical tasks. So what they did was play speech or music and a background noise and train the model to just, all it was doing was estimating the fundamental frequency of that speech or music. They trained lots of different networks, and then they picked the networks that gave sufficient accuracy in predicting the fundamental frequency. And then they performed different psychophysical tasks that are pitch related, for which there were human data. So again, I won't go through all the details, but they looked at a pitch discrimination task, a pitch estimation task, where they were looking at alternating phase harmonics. The pitch discrimination, they were looking at the effect of removing the lower frequency harmonics. And the human results are shown here, and the model results shown in the next column. And you can see that the patterns are very similar between the model and the human results. And they also looked at pitch-shifted complexes, either amongst the low frequency harmonics or high frequency harmonics. Again, human results and model results look very similar. They looked at pitch estimation if you're just mistuning one harmonic, and looking at tuning different harmonics, again, comparing human and model results. And then finally, looked at these interesting stimuli that are referred to as transposed tones, where they're really high frequency harmonic complexes that have temporal modulations that are a bit like low frequency tones. Again, I won't go through the details, but the pattern of the DNN predictions are very similar to the human results. And then what they did, they said was, well, what if instead of using this fixed cochlear model that's based on mammalian cochlea, what if we have the DNN try to learn a cochlear time frequency analysis to predict fundamental frequency? Does it end up learning something like humans? And I'll just do this summary slide here. They're showing here a correlation, so similarity between the human and model results. The black is showing with the fixed mammalian cochlea, and the magenta is showing what they get with different DNNs when they have to learn the cochlear filtering. And for 3 out of the 5 tasks, learning the cochlear tuning gives very different performance on these tasks than human performance. So it looks like the cochlear filtering is affecting how the DNN can learn and makes it behave like a human. And just another application, they've extended this to now do more complicated human tasks. And so this is a little small, but they played around with this cutoff frequency for the temporal precision. So instead of dropping at about 3 kilohertz, they said, what if we reduce the cutoff frequency so there's less temporal precision? And again, I'll just show the summary results. These are shown in the dashed lines here. Again, this is, it's on the right here. This is the similarity between the human and model performance shown here as a function of that cutoff frequency for the temporal precision for a bunch of sound localization tasks, voice recognition tasks, and work recognition tasks. And what you can see is as you reduce the cutoff frequency, at some point the model stops behaving like the humans, but the frequency at which it does that is different depending on the task. So in conclusion, the level of physiological accuracy required in a cochlear model to explain perceptual performance seems to depend on the exact task. Including the cochlear's nonlinear frequency scale seems to be the most important over a broad range of tasks, but maybe not all of them. The cochlear nonlinearity, so this compression suppression, auditory nerve fiber temporal precision, seems to be important for certain listening tasks. And I didn't show it here, but adaptation in the inner hair cell auditory nerve fiber synapse, and efferent feedback to the outer hair cells also seems to be important for explaining forward masking, but I didn't have time to discuss that here. So I just want to thank the funding sources and the many collaborators and trainees, past and present, in the lab contributed to this work, and thanks for your attention, and I hope we've got a few minutes for questions.
Well, questions?
Yes.
Very interesting talk. So, just not from this field,
Yep.
It's so interesting. And I just want to hear a little bit of clarification about, the roll-off effect in terms of the intelligibility is a function of the loudness, right? So you show the result, and they show how the model can predict of this previous collected data or similar to this.
Yeah.
Yeah. So, can you comment on maybe explain a little further how this, what's the actual mechanism in the cochlear is actually contribute to this function?
Yeah, so what's happening for the low sound pressure levels, it's this audibility effect. So at low sound pressure levels, there'll be, for the particular speech spectrum, there'll be parts of the speech, say in vowels, the low frequencies, or in consonants, fricatives, plosives, and higher frequencies, well, there'll be lots of energy, but it's distributed across frequencies, and so as you increase the sound pressure level, you'll start getting more and more auditory nerve fibers that are starting to represent that, but only a small population, and you have to go to a fairly high level before you're recruiting a large population of auditory nerve fibers to fully represent that. So part of it is recruiting enough neurons to have a faithful representation. They've got their own internal noise, their own stochastic spontaneous activity that limits how well one nerve fiber can encode things. So you need a population at each frequency, and so you need it to be loud enough to recruit enough auditory nerve fibers. And then this rollover effect is that basically what happens is these cochlear nonlinearities help stabilize the representation at around conversational speech levels, but then at a certain point the tuning becomes very broad. So if I go back to I'll jump back to this figure here. So as the sound pressure level gets higher, you get a bit of broadening of tuning. If you go to higher sound pressure level, so this is above conversational speech level, which is about 65 dBSPL, the tuning becomes super broad, and so the neural representation kind of spreads out and becomes really distorted. So the rollover effect is primarily that these cochlear nonlinearities, compression and suppression, help kind of stabilize things around 65 dBSPL, but as soon as you get above that, things start to broaden so much in the tuning that the neural representation becomes very distorted and not matching what you have at the lower sound pressure levels.
Just follow up very quick. So, is there any evidence showing, the signals passing through those cochlear into the brain, for example, auditory cortices, will also respond differently as a function of the sound pressure level?
Yeah, so there have been some studies. So I mean, there've been auditory nerve recordings looking at responses to speech and noise as a function of level, or you can also, so this post-mortem case is kind of the most extreme case you can have of impairing the outer hair cells, but if you also have more moderate damage to the outer hair cells, then you can just have partial loss of these nonlinearities. So if you look in animal models that have had ototoxic hearing loss or sound-induced hearing loss, at their neural representations at the auditory nerve level, you see this distorted representation, that these fibers that would normally respond to high frequency sounds are responding more to low frequency sounds than normal. And there's been some studies going up to higher centers, midbrain and cortex, showing that distortion tends to carry through as well. And then there's also plastic mechanisms where you can get distorted frequency scale in the cortex if the hearing loss is bad enough. So things are changing centrally as well. Daniel?
Yeah. Can you go back to the slide?
Which one, the speech one?
The rollback.
Sorry,
The rollback one. From
oh yeah.
I have a chicken and egg question about this. So,
Yeah.
So if you look at the 70 decibel, basically where the highest peak is.
Yes.
So I think speech and language pathologists would agree that an efficient way of articulation would be around 60, 70, 80
Yes.
decibels. And then below that you would strain your voice, and above that you would strain your voice.
Yes.
Do you think we have this peak because it's the most efficient way to communicate, or do you think it developed in tandem? Do you think that there's a link between speech production and speech perception based on this?
So again, so this peripheral model has been partially humanized. So we've modified the place frequency map, the frequency scale based on human rather than say cat place frequency tuning, and the middle ear filter is human in shape because we can do measurements of the human middle ear filtering, but all the other parts of the model are based on measurements from different mammals, different parts of the auditory periphery. And so that would argue that actually this is something to do, again, like I said, with the frequency scale and how that might have evolved, that it's actually got to do with how mammalian ears in general have evolved, and the sound pressure level of environmental sounds and animal vocalizations, nothing to do with our human vocal tract. And so it may be that, yeah, human vocal effort is evolved to produce vocalizations at the right level, but it seems like also animal vocalizations would then need to be kind of in this sweet spot here and matching, again, physiologically what's healthy for them in the speech production side of things. And then some of it's going to be kind of competing with background noise as well. So all of these predictions that I've shown here are in quiet. There's some data that Yuji's also done some predictions of, showing what happens if you add background noise. And basically these curves kind of shift down. You still get the rollover effect, but as you increase the background noise, the intelligibility drops. Excuse me. And again, the models, our model's able to predict that fairly well. That the Haspy model, the simpler cochlear model, doesn't predict that very well. It has weird changes in the rollover effect as a function of background noise level. So I think this has got to do with how the mammalian ear and brain have developed to deal with environmental sounds and animal vocalizations, and human speech is just making use of the same features, structure.
The question?
Yeah, you sort of did answer my question, but so when there's background noise, the rollover doesn't shift right, it just is suppressed, like the intelligibility is the only thing?
Yeah, so the slope of the rollover stays the same, and you just get a drop in the intelligibility.
Okay, so
and again, our model can predict that. The Haspy model, that's kind of a simpler cochlear model, doesn't seem to be able to predict that as well.
okay, so then there's essentially, if you have background noise and you're trying to do like a speech recognition task, for example,
Yes.
there's no point in increasing the intensity of your stimulus?
Exactly, yeah. So you get a benefit if you for a fixed noise level, you increase the speech, you get a benefit because the signal-noise ratio is increasing, but for a fixed signal-noise ratio, as you increase the sound pressure level, the intelligibility will actually drop.
Okay, and the sweet spot is still 60.
So the sweet spot's still around our normal conversational speech level.
Maybe I'll just ask a question, sort of following up. So with hearing loss, because I know
Yes.
you've done a lot of work
Yeah.
with hearing loss, so what happens when the person doesn't hear until a certain level, and
Yeah.
then there's a compression, extra compression that happens?
Yeah.
Because they have a
So
limited
again,
bandwidth.
we've got some predictions of that. I didn't think to put any slides in. This is some older results that we've got with earlier versions of the model. But basically, the human data and the model predict this pretty well, that basically unaided, so if the person's got hearing loss and isn't wearing their hearing aids, this rollover effect goes away and it becomes an audibility effect because of the damage in their cochlea. You need to present it at a higher level in order to make it audible to them. But as soon as you amplify the sound with the hearing aid to compensate for their hearing loss, the rollover effect comes back again. So people who have hearing loss but are hearing aided, so amplified through a hearing aid, they experience the same rollover effect as well. So it really does seem to be got to do with what's happening to the neural representation that's influenced by these audibility effects and cochlear nonlinearity effects and this broadening of filtering effects. Yes.
You showed a suppression around a probe tone.
Yes.
The probe tone stimulus is harmonically rich. Are there similar suppression regions surrounding each harmonic?
So very good. So that was what I was trying to convey with these tones. So if you have piano tones where we've got a set of harmonics for this low frequency tone, they're all mutually suppressing each other, and so what happens is that the higher intensity harmonics tend to win out. And so this is actually important for vowel perception. Again, we've done some modeling of this. I don't have any slides here to show it, but if you look at the representation of a vowel in auditory nerve fibers, it's the harmonic, the highest harmonic that's near a spectral peak, so a formant we refer to them in vowels. So these spectral peaks that come from the resonance of the vocal tract, they tend to dominate the neural representation and suppress the response to the harmonics nearby. So if you look in the auditory nerve representation and our model representation of vowels, you get these really strong responses to the harmonics at the formant peaks, and everything else tends to get really suppressed. So you get this extra stable representation of just those harmonics.
And if the whole spectrum shifts in sweeps, would you expect these to kind of dance around?
So yeah, it'll be a little bit, I mean, here you get this interplay, so natural sounds, the amplitudes of the different harmonics differ a bit, and so it depends a bit on the relative amplitude of these, and it depends on the width of tuning at that particular point. So again, in terms of absolute bandwidth, it's narrower at these low frequencies, which is why you can see kind of the neural response spatially much better for these lower harmonics, and then for the higher frequency harmonics, they become less resolved spatially because the absolute bandwidth gets broader, so things get smeared out. But they're all mutually suppressing each other, and so if one of them happens to be a bit higher amplitude, it'll tend to suppress the nearby ones with this extra complication of if it depends where you are on that kind of nonlinear frequency scale, which one will tend to win out, how much they get smeared out at the higher frequencies.
Okay, well, I think we'll stop there, and please join us out in the lobby for more talk, and Ian will be around to answer more questions.